The University of KwaZulu-Natal's College of Agriculture, Engineering and Science is committed to making a difference in our world, from the classroom and laboratory to the field. It provides one of the finest study and research environments on the African continent and is recognized internationally as a center of excellence. Agriculture, Engineering and Science is one of 4 colleges that make up the University of KwaZulu-Natal and is home to over 8,700 students and approximately 700 staff. 3 of the university's 5 campuses form the core of agriculture, engineering and science activities in the region. The college comprises 5 schools: agricultural, earth and environmental sciences, engineering, chemistry and physics, life sciences and mathematics, statistics and computer science. Engineering is offered on the Howard College campus, agriculture on the Pietermaritzburg, and all the other sciences on both the Pietermaritzburg and Westville campuses. Exceptions are agricultural engineering, which is offered in Pietermaritzburg, and geology, which is offered at Westville only. To compete with the best and to meet the challenges of the 21st century, South Africa needs highly skilled professionals in the areas of agriculture, engineering and science. We offer a wide array of interesting and innovative degree programs that open doors to a variety of different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree and one of only 2 universities in the country to offer a BSc in land surveying. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science, and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural disciplines for a 4-year Bachelor of Science in Agriculture degree. These are more career-orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the envy of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agricultural engineering. Adjoining Ukulinga is the Bisley Valley Nature Reserve, which supports a diversity of plant and bird species, as well as some large mammals. It provides a rich learning and research environment for staff and students. Based on the Westville campus, the college possesses unique facilities for research into higher voltage power transmission. These facilities are available in only 3 other places in the world and form part of the ESCOM-sponsored Science and Technology Innovation Park. The Science and Technology Education Center showcases the college's scientific achievements and promotes the public understanding of science, engineering, and technology within the region. The college houses a wide range of sophisticated equipment in its microscopy and microanalysis units, staffed by highly qualified and skilled technical specialists. The College of Agriculture, Engineering, and Science prides itself on its graduates who are highly sought after. In a market characterized by a shortage of qualified professionals in science and technology, our graduates are readily employed around the world in the fields of agriculture, science, commerce, industry, engineering, and research, to name just a few. Research plays a major role within the college and operates at the cutting edge of technology. The college has the highest research output in the university and produces the most research publications. There are over 60 well-established research groups and centers within the college. The African Center for Crop Improvement caters for students from all over Africa. It is one of only 2 African plant breeding programs on the continent. Based in the School of Agricultural, Earth and Environmental Sciences, the African Center for Food Security focuses on alleviating hunger in Africa. It offers food security training programs, undertakes academic and applied research, and provides policy and program support. Promoting sustainable engineering practice is the Center for Research in Environmental, Coastal and Hydrological Engineering, or CRESH. It deals with the interaction between engineering and the natural environment. The Mechatronics and Robotics Research Group in the School of Engineering provides an excellent research and education environment for its senior undergraduate and postgraduate students. Judged one of the best research centers at the university, the Center for Plant Growth and Development focuses on high-quality research in the plant-based disciplines of plant physiology, plant molecular biology, and ethnobotany. The Center for Quantum Technology deals with physical phenomena at the microscopic level. Quantum physics led to the development of the transistor and the laser, and is currently being used to develop quantum computers and quantum cryptography. The college is committed to access and readdressing the disadvantages and inequities of the past. The highly successful Unite Engineering program and the Science Access Foundation and augmented programs offer alternative access to university, equipping students from disadvantaged backgrounds with the skills and knowledge to pursue their degrees of choice. Both these programs contribute significantly to advancing Black African scientists and engineers in South Africa. Staff and students contribute to a number of outreach and community initiatives that focus on social responsibility, sustainable development and upliftment of the wider community. The college also engages in activities involving the pursuit and understanding of science, engineering and technology through exhibitions, festivals and roadshows that target high school learners and the general public. The college has minimum entry requirements for its programs. These differ from degree to degree, but all require a national senior certificate and minimum performance levels in mathematics and either physical or life or agricultural sciences. For those who do not meet the minimum requirements, access programs are available. Additional information and brochures can be obtained from the contact information accompanying this video. The College of Agriculture, Engineering and Science is proud of its contribution to the university, the region and the country.
UKZN is one of the best. I definitely understand it's one of the best in South Africa. So for me, there was no other institute that I would have preferred other than UKZN.
We have the best facilities here in terms of laboratories, in terms of equipment, in terms of human resources, in terms of literature.
UKZN is one of the best universities in the country that teach agriculture or natural sciences.
What I think is good about this college is the level of international collaboration that we have. We have friendships with universities all over the world, which means that we're keeping up to a great international standard.
UKZN has got much more facilities and it's recommended worldwide to do your science studies here. Life begins here. Yes.
We continually strive to be academically excellent, innovative in research, and critically engaged with society. We invite you to join us or work with us to be part of this exciting team.
Good morning, students, and welcome to session 3 of the Matric Helpline for Life Sciences. I hope you're all doing well this morning. Thank you for joining in. And today we're going to be looking at the topics DNA and meiosis. Okay, I'm just going to connect you to my PowerPoint. If you can give me 1 minute. Okay, can you see my PowerPoint screen? Just give me a thumbs up, or you can just shout out if you can see that. Yes. Okay, perfect. Okay, great. So I hope your test went well. Those of you who did a genetics test the other day, I hope that went well. Any feedback quickly? Any students who are here from Thursday?
I don't see Tessie online as yet.
Oh, all right. Yeah. Okay, so let's start with our session for today. Okay, so we are doing DNA and meiosis today, and the subtopics that you need to know for these two topics are particularly DNA structure, function, and location. Okay, so you need to know how DNA is structured, what are its monomers, how DNA is shaped, how does it maintain that shape? And the function of DNA is to do what? Can anybody quickly tell me what's the function of DNA? I'm starting with a very easy question. Anybody? What's the function of DNA?
It carries genetic information.
Yes, that is correct. Okay, so it controls the functioning of the cell because it contains the genetic information that defines that cell. Okay. You need to know DNA, the process of DNA replication. Okay, how DNA makes a copy of itself, and it's an identical copy of itself. You need to know about DNA profiling. Okay, and you need to also know about RNA and the differences between DNA and RNA. And with RNA, you need to know protein synthesis, the process of protein synthesis. Okay, and then for meiosis, you just basically need to know all the stages of meiosis, exactly what happens in each stage. Okay, so I expect you to get full marks for the meiosis section. It's the easiest section of your whole paper. So you don't lose marks in the section. It's very easy to learn off. Okay, so please make the most of your meiosis section. Okay, before I continue, I'd just like to remind you that my colleague, Dr. Nimisha Singh, will be posting links on the chat. If you can please click on them to register. And also, if you want the previous videos of this year's sessions or last year's sessions, you can access them on the website, which Dr. Singh will put a link for you there in the website, to the website. And if you can post any questions that you might have for me or Dr. Singh, you can post them on the chat and she will address them on the chat. Or if you need to ask me something, you can also just put it on the chat or even just shout out, it's fine. Okay, so let's continue. Okay, so we're jumping straight into it and we're starting off with our definitions. Okay, so let's give the biological term. Okay, you're going to shout out to me or you can put it on the chat. Okay, so give the biological term for monomers of nucleic acids. What are the monomers of nucleic acids called?
Yeah, but you'll put the stuff away.
Yes, Rob. Carry on. Good, that's it. Should we want your way?
Students, anybody? Okay, you got to take a try at these things so that you know whether you know them or not, because this will be a section in your paper. Okay, what are the monomers of nucleic acids called? Just take a try. Cam, you have an answer?
Nucleotides.
Yes, that is absolutely correct. Well done. Okay, then what is the process by which DNA makes an identical copy of itself? Super easy.
DNA replication.
Yes. Okay, parts of the DNA molecule that code for the production of amino acids. Okay, this one's a little bit tricky. Okay, so it's actual parts of the DNA or the chromosome that code for the production of amino acids.
A gene.
Yes, okay, that's also correct, but the actual answer is an exon. Have you heard of that word before?
No, ma'am.
Okay. And Nimisha, do you want to explain that?
Okay, so in your chromosome, you have genes. Right now, our genes is our coding region with our nucleotides, our adenine, guanine, cytosine. And what's the last one I missed out?
Thymine.
Yes, thymine. Out to the students.
Oh, sorry.
It's okay, Jerusha. Okay, so we have those four, right? Now, through evolution, right, you know, as the organisms got more complex, the different genes that code for the specific amino acid chains to make our proteins has also evolved over time. So as the proteins got modified, as the organisms got more complex, some of the coding region of the gene wasn't necessary anymore. However, they stay within your actual gene. So this is where we have the terms exons and introns. Exons is the sequence of our nucleotides that actually code for your actual mRNA transcript that will then be translated to protein. Now, each gene can have several exons, but in between your exons, you're going to have what is known as introns, which is your non-coding region. So they are also sequences of your nucleotides. However, those parts are not translated. However, they are during transcription, those parts need to be cut out to have your transcript for translation. So for you to remember, exons are the coding region of your gene, while introns are the non-coding region. Q?
So ma'am, are introns like non-coding DNA?
Yes, non-coding. So if you're to look at a sequence of a gene, you're going to have some nucleotides in a sequence in capital letters and some in small letters. So the capital letters indicate coding region, while the lowercase letters of your nucleotides indicate the non-coding region, which is your introns.
Your introns are also sometimes known as junk DNA. So it's just there,
ma'am,
but it's not providing any function. And there's quite a lot of junk DNA in between your exons. Okay. Okay, is that fine? Thank you, Nimisha. Okay, so small proteins that help to wrap and condense DNA.
Histones.
Yes, correct. Okay, structure that joins the chromatids of a of a chromosome.
Centromere.
Yes. Okay. Very good. Okay. Then what is the term for a triplet of bases found on a tRNA molecule?
Nucleotide.
No. A nucleotide is a single base. What is a triplet of bases found on a tRNA molecule?
Is it an anticodon or a codon?
It's a, you tell me. You tell me what.
Anticodon.
Yes, okay, it's an anticodon. What has the codon?
The mRNA.
Yes, that is correct. Okay, so next, what is the process by which mRNA is built on a DNA template?
DNA transcription.
Yes, okay, it's transcription. Okay, the process whereby different amino acids are assembled to form a specific protein.
Translation.
Yes. Okay, what is the bond joining amino acids in a protein called?
Polypeptide bond.
Sorry,
Polypeptide bond.
Yes, that's correct. Okay, but it's actually a peptide bond, and then many peptide bonds is a polypeptide chain. Okay, you with me?
Yes.
Okay. What is the site of protein synthesis in the cytoplasm?
The ribosome.
Yes. Okay. Okay, good. So everybody knows their biological terms. Well done. Okay. Oh, there's one more. Okay, so what is the point at which crossing over takes place?
Is it chiasma?
Yes, correct. Okay, what is the division of the nucleus called? This is also a tricky one. Division of the nucleus.
I think it's kerokinesis.
Yes, very good. Okay. What is failure of one or more chromosomes to separate during meiosis? What is the term for that? It's what?
Disconjunction.
No, I haven't heard of that one before, but it's actually non-disjunction. Okay, you got that. Okay, then what is the organelle that forms the spindle fibers in animal cells?
Centriole.
Yes. Okay, chromosomes other than sex chromosomes?
Autosomes.
Autosomes, very good. Okay, yeah, so you are doing well with the biological terms, and it's a good section to try and get full marks. Okay, because I think each of them are worth 2 marks each. Okay. Okay, so now let's go into some short answer questions. Okay, when a thief broke into a car, he cut his arm on the broken glass. Scientists extracted DNA from the blood found in the broken glass. They analyzed this DNA sample and compared it to the DNA from 3 suspects, P, Q, and R. The table below shows the results of the analysis of the DNA from each source. Okay, so the blood from the broken glass, you can see all those bands there. Those are segments of DNA, okay, that are separated out. Okay, so you can think of each of those bands as a section of DNA, okay? And you can see the same for P, Q, and R, okay? So they took DNA samples from P, Q, and R and compared it to the DNA sample that they got from the blood on the broken glass. Okay, so what do the diagrams above represent?
DNA profiling.
Yes. Okay, so which suspect is most likely the thief? Okay, so what are you looking for? You're looking for the bands that match. Okay, so the 2 samples have the same DNA. Okay, so which 2 bands match? The blood from broken glass matches which P, Q, or R?
Q.
Q, very good. Okay, you can clearly see that all the bands correspond between the 2. Okay, give a reason for your answer above. Okay, we said that the bands correspond with each other. You can see that the first band on the top there from the blood from broken glass is also found in Q sample. And all of those bands are exactly the same as suspect Q. Okay, say 2 possible disadvantages of using this evidence in a court of law. So why is DNA evidence a little bit difficult to use in court? Anybody want to take a try?
Because DNA profiling is done by humans, therefore it could be due to human error and they could mix it up.
Yes.
Wrong person
Mix what up?
Mix up their DNA.
Okay, very good. That's a good answer. Any other people want to add to that?
Innocent people may be framed.
Yes, very good. Because they can plant DNA on the scene. What else? Are there any answers in the chat?
No, no one's posting in the chat today. If
Oh,
you're feeling shy, you can post it on the chat and I'll read the answer out.
Okay, also suspect Q could have a twin brother, okay? So their DNA profiles would be the same.
We have suspect Q and a Q student attending.
That's a okay. There's only a small amount of DNA used, so that's not really reliable, okay? So those are your possible disadvantages. Okay. Okay, let's move on. Now you can see the diagram on the left. What is that showing you? A picture of a ribosome with what inside?
mRNA.
Yes, and what's that funny pin-looking thing?
tRNA, the NZ codon that will bring the required codon to the strand of mRNA.
Very good. Okay, so name organelle X. What's organelle X?
Ribosome.
Yes, very good. Okay, molecules W and Y. What's W?
With mRNA.
Okay, and what's Y?
tRNA.
Yes. Okay, and name the monomer of molecule W.
DNA.
No. We said molecule W is RNA, so what is the monomer of mRNA?
Nucleotide.
Yes. Okay. Where in the cell is organelle X found?
In the cytoplasm.
Yeah, yes, that is correct. And molecule W, where is it formed?
Chromosome.
Where?
Chromosome.
No, try again. Where is it formed? So where is mRNA made?
In the nucleus.
Still have
Sorry?
two boys, such a big boys, not small children. How they help her sit in making videos and support her. See, this is her son.
Sorry, what's the answer? Nucleus. Okay, can I just ask you to mute if you're not answering, okay? Okay, next. Insulin is one of the hormones responsible for the control of blood glucose levels in humans. It is made of two long amino acid chains, A and B, which are joined. Chain A is made up of 21 amino acids. And can I ask you just to mute? Students, can you all mute yourself?
No, this lady, is more talented. But still, she has more followers, she get more money.
Okay. Sorry about that. Let's move on. Okay, so chain A is made up of 21 amino acids and chain B of 30 amino acids. The diagram below represents the amino acids present in each chain. Okay, so you can see here chain A and chain B, they are joined together, and this represents your insulin molecule. Okay, so name the process whereby insulin is produced in a normal human cell. Students, any ideas? Insulin is a protein. You've got 2 polypeptide chains there. So what's the process whereby insulin is produced? I gave you a clue. Any ideas? It's just throwing you off because it's saying insulin, but insulin, they've given you the what it looks like. It's protein. Okay. So name the process whereby insulin is produced in the cell.
Is it protein synthesis?
Yes, correct. Okay. Identify bond X. So the one between the 2 nucleotides there. Sorry, amino acids.
Peptide bond.
Yes. How many nitrogenous bases in a DNA molecule code for the amino acids in chain A? You've got 21 amino acids in chain A. So how many nitrogenous bases in the DNA molecule code for that?
62.
Sorry?
It is 62, ma'am.
Not 62, it'll be 63. Okay, so it's going to be 21 times 3 because it's 3 nitrogenous bases per amino acid. You with me?
Yes, ma'am.
Okay. Okay, the table below shows the mRNA codons that code for some amino acids. Okay, you can see there, you've given mRNA codons and the corresponding amino acid. Name the nitrogenous base represented by G in the mRNA codon CAG. Okay, so what's CAG? Use your table and read it off there. What's the amino acid that CAG codes for?
Glutamine.
Yes, correct. Okay, read the chains from left to right and give the codon for the 4th amino acid in chain B. Give the codon for the 4th amino acid in chain B. So what is that? Can you see that? It's GLN. So what's the codon for GLN?
CAG.
Correct. The DNA-based triplet that codes for the 1st amino acid in chain A. So that is GLY. It's asking for the DNA-based triplet that codes for the 1st amino acid in chain A. So you've got the mRNA triplet. What's the DNA triplet? Okay, so first tell me the mRNA triplet for GLY.
It's GGA.
Correct. Now, what is the DNA triplet?
CCU.
No, we're talking DNA. So we don't have a U, we have a T. So it'll be CCT. Okay. Is everybody with me on that one? They will oftentimes give you a question like that where you have to work from mRNA to DNA or DNA to tRNA. Okay, you have to be able to go from codon to anticodon to DNA sequence. Okay, you got to be comfortable with that. Okay, give the codon for valine. Give the anticodon for valine. Okay, so you've got the codon. What's the codon for valine? Students, what's the codon? Yeah. What's it?
GUA.
Right. Now, what's the anticodon? What's the anticodon for GUA? So we're looking at what's on the tRNA molecule.
I think it's CUU.
No, try again.
CAT.
No, T is not going to be on your RNA, remember? Instead of thymine, we have uracil. So what is the anticodon for GAU? Sorry, GUA.
It's CAU.
Correct. Okay. CAU. Okay. Now, a section of DNA molecule has the following base sequence: CTTACA. Name the nitrogenous base represented by C in the DNA molecule. What is the nitrogenous base represented by C? What is C called?
So what is the nucleotide's name for C?
Cytosine.
Yes, correct. Okay, then the percentage of guanine in this DNA molecule is 30%. Give the percentage of thymine in the same molecule. Okay, so it's telling you the percentage of guanine. What, do you see a G there? No. So what pairs with G?
Cytosine.
Right, so you can see two cytosines in that sequence. Okay, so both your cytosines in that sequence, now that represents 30%. Okay, so now it's asking you, give the percentage of thymine in the same molecule. If two C's are 30%, what is two T's? You can see two thymine there. So what's the percentage?
Do you times it by 2 because there are 2 T's, so it'll be 60%?
No. Remember we said that the percentage of guanine is 30%. Okay, but there's no guanine, there's cytosine. And that obviously, that pairs with guanine. So the amount of guanine and cytosine are going to always be the same because they pair with each other. Okay, so we've got two C's in that DNA molecule, and both of that represents 30%. Okay, so we've got two C's equals 30%. So 2 T's equals how many percent? It's very easy, don't overthink it.
Ma'am, is it 60%?
No.
Ma'am, I have a question, ma'am.
Yeah.
Won't the adenine also combine thymine since there's also 2 A's?
Yes, but it's not asking you for that. It's asking you only for thymine. It's not asking you for adenine as well. And it's only asking you for the percentage in that sequence, not in the other strand. The thymine that pairs with adenine there will be in the other strand. So it's not asking you for that. It's just asking you for the percentage of thymine in the same molecule. So just those 6 letters. So 2 cytosines equals 30%. 2 thymines equals how many percent? You're all overthinking it. It's very easy, guys. So write this down as I'm saying it, then you'll see it. 2 cytosine equals 30%. 2 thymines equals X. It's the same proportion, okay? So that DNA molecule has the same proportion of cytosines as it does thymines. There's 2 cytosines and there's 2 thymines. So it's the same proportion of that DNA molecule. And if that proportion is 30%, it means that thymine is also 30%. Okay, so the proportion of cytosine is 30%. And there's also 2 thymines there. The proportion of cytosine and thymine are the same. So therefore, thymine is 30% of that molecule. Are you with me? Are you kicking yourself for overthinking it? And it's just for 2 marks. So it's something that doesn't require a lot of working out. Okay. Dr. Singh has put that in the chat for you. Yes, Fahima, you got it correct. It's 30%. Okay. Give the mRNA sequence from left to right for this segment of DNA. Okay, so what's the mRNA sequence?
Is it GUU?
No. We're talking DNA. Sorry, you are right. Give the mRNA sequence. Sorry. Yes, you are correct. So the mRNA sequence, you are correct in saying uracil instead of thymine. Okay, so just repeat that for me.
GUU.
Yes, correct. Okay. Thanks. Okay, so the table below shows the DNA triplets that code for some amino acids. Give the sequence of amino acids that would be coded for by the section of DNA above. So what amino acids does this DNA code for?
I think it's glutamic acid and cytosine.
Yes, cysteine. Okay, so glutamic acid and cysteine. Okay, those first two DNA triplets there. Okay, give the anticodon for the amino acid methionine. So what I want you to notice, okay, just as a rule of thumb, your DNA transcript is the same as your anticodon or your tRNA transcript, except that instead of thymine, you have uracil. However, your mRNA transcript is corresponding, complementary to your DNA strand. Okay. So your DNA strand is complementary to your mRNA, and your mRNA strand is complementary to your tRNA. So your DNA strand and your tRNA strand are the same, except instead of thymine, you have uracil in your tRNA. Okay. So give the anticodon for the amino acid methionine. So here's the DNA. Okay, so you're going to go from DNA to mRNA to tRNA. Okay, so write it down there. What's the DNA triplet for methionine? TAC. Okay, so now what is the mRNA for TAC? What's the mRNA, students, for TAC?
Is it AUG?
Yes. Okay. Now, what is the anticodon? So, what is the transcript for the tRNA?
UUC.
No.
The complement of uracil?
UAC.
UAC. Okay, that is the tRNA. So now what do you notice between the tRNA and anticodon and the DNA? It is the same. The only difference is that instead of a T, you have a U, because it's RNA in the anticodon. You with me? If you look at the chat, you'll see it there. Dr. Singh has typed it out for us. Okay, so look at the DNA, it's TAC, and the anticodon is UAC. So it's the same, it's just that instead of a thymine, you have a uracil. Okay, so that is an easy way to remember it, because they definitely gonna ask you to go from DNA to mRNA to tRNA and everything in between. So they can ask you to go from, like this question now asks you to go from DNA straight to tRNA. So you, to save time, you must know, okay, it's the same as the DNA strand, just instead of T, it has U, because it's now a tRNA strand that the question is asking for. Okay, so it's RNA, so it can't have thymine, it must have uracil. Okay, so let me repeat it. Your mRNA strand is complementary to your DNA strand. Everybody got that? Give me a shout out, yes. Hello? Students, are you with me?
I don't think the students had their breakfast this morning.
Nobody had breakfast. It's too early. You with me?
Put a thumbs up if you understand. Oh, Siada said yes, ma'am.
Thank you, Sianda. Right. So your DNA is complementary to your mRNA. And your tRNA is complementary to your mRNA. Therefore, your DNA and your tRNA are the same, except that instead of thymine in your DNA, you're going to have uracil in your RNA. Okay. So you need to be able to go straight from DNA to tRNA. And you can do that because the tRNA is the same as the DNA strand, except that it has uracil instead of thymine. I hope that's very clear now. Okay. Okay, let's move on. Okay, the diagram below shows the karyotype, and can somebody just confirm whether you are doing karyotypes in school? These were in past papers. I just wanted to find out if you're still doing karyotypes. Did everybody learn karyotypes? What it is? Anybody? Anybody can just confirm that they know what they are looking at here on the slide. It's karyotype. Have you heard of this before?
Q.
Okay, thanks. Okay, so you do this still. Okay, because these are from past papers. Okay. Right, so the diagram below shows the karyotype of an individual with a disorder caused by non-disjunction. Let's remind ourselves, what is non-disjunction?
When the chromosomes fail to separate itself.
Yes, okay, so if they fail to separate, it means there's going to be an extra copy going into the one cell. Okay, in anaphase, remember, those chromosomes separate to each side of the cell. They separate and go towards the poles. So if one chromosome does not separate, it means that there's going to be two copies of it on one side of the cell. Okay, which means that that is going to end up in one cell. So one cell will have two copies of that chromosome. Okay, because they did not separate during anaphase. Okay, so name this disorder that you can see there in the karyotype. What can you notice? Where's at which point is the non-disjunction? Which chromosome?
21.
21. So we have an extra copy of chromosome 21. What is that disorder called?
Down syndrome.
Yes, correct. Okay, the phase in meiosis when non-disjunction occurs. I just told you now. It is what?
Yanda, your mic is on.
What is the phase in meiosis when non-disjunction occurs? I just told you. Which phase does the chromosome not separate?
Anaphase 1.
Yes, okay. The type of mutation that is a result of non-disjunction? Anybody?
Ma'am, is it aneuploidy?
Yeah, but there's another term for when you just have one chromosome. But yes, that is a general term that you can use. You may get that mark. Any other suggestions? Okay, we'll go with aneuploidy for that one then. Identify the type of chromosomes numbered 1 to 22.
Autosomes.
Yes, correct. What is chromosome 23?
Monosome.
Yes, correct.
Could you please explain what the other answer could have been for 1.5.1c?
Okay, let me just find it for you. And Nimisha, do you have any other answer for that?
Sorry, my mic was muted. No, for other aneuploidy, I mean, some texts would just call it a translocation mutation. They don't give the specific.
Yeah, but
yeah,
it's
name.
the general term for an extra chromosome. So that would be aneuploidy. Yeah, that's an acceptable answer. Okay. Okay, next. The diagram below represents some nucleotides in a single strand of DNA. Give the letter of the part that represents A. Sugar molecule. Which part represents a sugar molecule?
I think it'd be Y.
Yes, that's correct. And which one represents a phosphate molecule?
X.
Yes. Okay, how many nucleotides are represented in the diagram?
4.
Correct. Write down the nitrogenous bases from top to bottom as indicated by the arrow. Write down the nitrogenous bases of the complementary DNA strand of this molecule. What is the complementary DNA strand?
I think it would be ACTG.
Correct. Okay. In that direction because it says follow the arrow. Okay, name 2 processes that require the 2 strands of DNA of a DNA molecule to separate into single strands as shown in the diagram. So what 2 processes require single strands of DNA?
I think it's DNA replication and and transcription.
Which is the process of?
Protein synthesis.
Yes, that is correct. Okay, so your DNA replication requires a single strand of DNA and protein synthesis also requires a single strand of DNA. Okay. Okay, next. Okay, now hemoglobin is a protein found in blood that carries oxygen to all the cells of the body. A portion of this protein is called a beta chain. If the sequence of amino acid in this chain changes, then a different form of hemoglobin called hemoglobin S is formed. Hemoglobin S cannot transport oxygen as efficiently as normal hemoglobin. Okay, so if you look at that table, you can see that there's a chain of amino acids. And what is the difference between normal hemoglobin and hemoglobin S? What can you see is the difference there?
The protein at number 6.
Yes.
I would say.
Instead of glutamine, it has valine. Okay, so there's just one amino acid that is different, and that now causes hemoglobin S to be formed, and hemoglobin S cannot efficiently transport oxygen. Okay, the table below shows the DNA-based triplets coding for the same coding for some amino acids. Okay, give the DNA-based triplet for amino acid 3. Leucine. So what is the DNA-based triplet for leucine?
GAC.
Correct. The mRNA codon for amino acid 4. Threonine. What's the mRNA codon? Remember, the mRNA codon is going to be complementary to your DNA strand.
TCU.
mRNA. Everybody seems to keep forgetting this. mRNA does not have thymine. It has uracil.
Sorry, ma'am. It's UCT.
Correct. No. You're telling me a
Oh.
T again. You're telling me T.
CU.
Yes, that is correct. Okay, so what is a change in the sequence of DNA-based triplets called? When you have a change in the sequence of DNA-based triplets. What is it called? What happened to hemoglobin S? Just one base changed. So what happened to, what is that process where one base changes and you've got a different thing?
Is it a mutation?
Yes, correct. Use the information in the tables to explain how a change in the sequence of the DNA-based triplets results in the formation of hemoglobin S rather than normal hemoglobin. Okay, so what is the base, the DNA base for GLU? Glutamine?
CTC.
Okay, and what is the DNA base triplet for valine? Look at the table.
CAC.
Okay, so what is the difference between CAC and CTC? What is the difference between those two things? What happened there? Instead of T, you had A. Okay, so one amino acid was incorrect. Okay, and that is how we got hemoglobin S. Okay, Does anybody know what type of mutation that is? That is a point mutation. Okay. Describe how a person with hemoglobin S would be affected. So now they've told you that hemoglobin S cannot transport oxygen as efficiently as normal hemoglobin. So describe how the person with hemoglobin S would be affected. They would be, if their body can't carry oxygen efficiently, if their cells can't carry, red blood cells can't carry oxygen efficiently, it means they're always going to be tired, they don't have enough energy, they're a bit slow because they don't have enough oxygen to the brain. Okay, are you with me? Students, are you with me?
Okay, so in the chat, I said that hemoglobin S causes the disorder sickle cell anemia, right? So anemia always means a lack of your blood being oxygenated. So, so how would a person be affected by hemoglobin S? Because of the shape of hemoglobin S, what happens to your blood cell, instead of being its circular biconcave shape, because of the single point mutation, because of valine being incorporated, your hemoglobin does not keep its original shape. Therefore, your blood cell is going to fold on itself, creating like a sickle shape of the cell. Therefore, you have the name of this disorder named as sickle cell. So it looks like a C or a sickle, a C-shaped knife. That's how it got its name. And because of the reduced surface area, your hemoglobin is unable to get oxygenated efficiently. And because of this lack of oxygenation, when these hemoglobins are transported throughout the body, the rest of your cells and your muscles and your brain, as Jerusha has mentioned, cannot be oxygenated efficiently. Therefore, you will be tired all the time, as well as many cellular processes are going to be unable to perform at its maximum. Therefore, you will have this disorder.
Okay, I hope everybody got that. Thank you, Nimisha. Okay. Let's move on to the next one. Okay, another karyotype. Okay, so the karyotype below represents the chromosome of a person. Give the collective term for the chromosomes numbered 1 to 22. We had this question already.
Autosomes.
Yes. State the gender of this person.
Female.
Why?
She has 2 X chromosomes, that's number 23.
Correct. Okay. Give one observable reason for your answer in 2.3.2. Okay, sorry, we just answered that. There's 2 X chromosomes at chromosome number 23. Okay. State Mendel's principle of segregation. Okay, what is Mendel's law of segregation? See, can you see that sometimes genetics comes into a normal DNA question? Okay. So you have to be prepared. Like even the question we had on insulin, you may be thinking now, okay, human responses, or I mean, sorry, endocrine section, but it's actually the DNA section. Okay, so don't get confused. They might incorporate other sections into the same question. Okay, state Mendel's principle of segregation. You must know this because it'll definitely come up, could even come up in multiple choice give the biological term, and now it's in a short answer question. Okay, so state Mendel's principle of segregation. So segregate means to separate. Okay, so the factors or alleles that control a specific characteristic, okay, they separate during meiosis so that only one copy of each allele or each factor is present in each gamete. Okay, so each gamete has a copy of one factor because the factors separate during meiosis. Okay, that is Mendel's principle of segregation. Okay, you got to learn that off. Describe how the karyotype of a person with Down syndrome would differ from the one above.
No, nothing. Thank you. No, thank you. There would be an extra chromosome at chromosome 21.
Yes, that is correct. Okay, that's correct. Okay.
Ma'am, I have a question, ma'am.
Yes.
Why is the extra chromosome always on 21 and not on any other chromosome?
Yes, that is the particular chromosome that just does not separate.
Thank you.
It's probably to do with the particular shape of that chromosome.
Thank you, ma'am.
Okay. I don't know if you have anything to add to that, Minisha. It's just
No,
that's the chromosome that just doesn't separate.
Yeah. Specifically for Down syndrome.
Yeah.
That's the main chromosome that is associated with that syndrome occurring.
Yeah. If you had any other chromosome that didn't separate, it would be another syndrome. It would be a different disease. Okay. Are you with me?
Yes, ma'am.
Okay. Okay, let's go on. Okay, the diagram below shows protein synthesis. Identify organelle V.
Ribosome.
Okay, that is correct. Organelle Q, molecule Q.
tRNA.
Where's molecule Q? tRNA, yes. Write down nitrogenous bases for codon R if it codes for glycine. Okay, so this is if you, oh no, it's given you there. Write down nitrogenous bases for codon R if it codes for glycine. Okay, so they've given you, there's a little tRNA floating around there with glycine, and what is the codon now for that glycine? So they've given you the tRNA. So they've given you the anticodon. Now it's asking you what is the codon?
CCU.
Correct. Okay, that is correct. Write down the sequence of the first two amino acids from left to right, which codes for molecule Q. Okay, so what's the first two amino acids from left to right? The sequence of the first two amino acids from left to right, which codes for molecule Q. Okay, so you've got your mRNA strand there. Okay, so we're going left to right. So we're starting with U, UAU, GGU. Okay, write down the sequence of the first 2 amino acids, which codes for molecule Q. So what is the tRNA now? What's the tRNA for that mRNA strand? Hello? So we've got UAU. What's the tRNA for that? Students, what's the tRNA for UAU?
AUA.
Right, AUA. So which molecule is going to go fit there? Which amino acid? There's your amino acids floating around there.
Isoleucine.
Correct. Okay. Then you've got GGU. So what is the tRNA for that? The anticodon for that.
It's
CCA.
CCA, so it's proline.
Correct. Okay. Okay, so what codes for? Oh, it just says write down the sequence of the first 2 amino acids from left to right, which codes for molecule Q. Yes, I did go left to right. Okay, Namisha, do you have any other understanding of this question? Because I'm getting isoleucine and proline.
That is what I also got as well. And I don't remember the paper giving them the actual table for threonine, because I mean they're not expected to know the codon for it. Anticodon.
Yes, yeah. What was
because R would have been
glycine?
Yeah. Then the next for now, because then the anticodon for threonine is on the table.
Okay, but it still says write down the sequence of the first 2 amino acids. So I would just write that down. Like what we did now, we said A, U, A, Q. You have an answer. Q, raise your hand. You have an answer.
Yes, ma'am.
Yeah.
How do these 2 amino acids code for Q? What's the correlation?
Yes, that's what we're just trying to understand, because it's not. It's actually coding for isoleucine and proline. Okay, but I would just write down the tRNA or the anticodons for AUA, GGU, because that's what the question asks. It says write down the sequence of the first 2 amino acids. Okay, from left to right, which codes for the molecule Q. Don't know why it's coding for the molecule Q though. But I would just write the anticodons for AUA and GGU. Okay, unfortunately, we don't have the model answer for this paper. So I'm not too sure.
Yeah, because it might be a typo, because threonine recognizes either ACA or ACC or ACU on the mRNA transcript, which this thread does not have. Let me just double check. This is paper 1. Oh, this was the comment test that they wrote.
Yes.
Question 2 and 3.
Okay, while you're looking for that, let's move on to the next question. But as I said, students, I would just write down the sequence for the first 2 amino acids. Leave it at that for now. Nimisha will just try and see if we can get the model answer for that question. Okay, let's move on in the meantime. Explain the effect on the protein formed if all guanine bases in molecule Q were replaced by cytosine bases.
Okay, so we were correct because the question said write down the sequence of the first 2 amino acids.
Yes.
From left to right. So if we did that, left to right is proline and isoleucine and proline, which is in the model answer. So I think it was a typo about Q.
Okay.
Yeah, they're supposed to say P because if you look at the image, the whole strand is coming from, P. So I think they made a mistake.
Oh, okay, okay. That makes sense if it was P. Okay, so explain the effect on the protein formed if all guanine bases in the molecule Q were replaced with cytosine bases.
So here it's just you explaining on the thread itself. If you replaced all guanines with cytosine, then how would your tRNA change?
Okay, so would proline be able to fit there anymore? Where you've got your guanines on your mRNA? It would not fit because now your CCA would not be complementary to that mRNA strand anymore. Okay, so if your G's were changed to C's, it would have a different amino acid being laid down there. Okay. Oh, sorry. Let me undo whatever I just did. And my slides have disappeared. Sorry.
The blanks got inserted.
Oh, blank. Okay. Okay, we're back. Okay. So we're saying that if G's were replaced with C's, would the same amino acids be able to fit there or be laid down there by the tRNA? No, it wouldn't, because the tRNA is an anticodon, carrying an anticodon for that mRNA. So if the mRNA strand changes, then the amino acids that the tRNA is carrying is also going to change. Okay, so you're going to have a completely different protein strand being formed. Okay. So now tabulate 2 structural differences between molecule P and Q. 2 structural differences between molecule P and molecule Q.
So molecule P refers to the DNA, and then your molecule Q is the mRNA. So there is a typo on the test question itself.
Okay. Okay. So what is the difference between mRNA and DNA? You must know this. They definitely going to ask you this. Okay, what is the difference between RNA and DNA?
Right, based on its structure, you give differences. Also, when they say tabulate, make sure you draw a table because you will get a mark for drawing a table.
For having a table. Okay.
With headings.
Yes. So you'll draw your table, your heading will be on top of the table. It'll say table 1 showing structural differences between DNA and mRNA. Okay, so this is very easy. You should all know it. Tell me 2 structural differences.
Molecule P is double-stranded. Molecule Q is single-stranded.
Correct.
And
What
molecule P has thymine, but molecule Q has uracil.
Yes, very good. What else?
DNA is deoxyribose and RNA is ribose.
Yes, very good. What else about the structure?
DNA is longer than RNA.
Yes, very good. Remember, DNA is in a double helix and mRNA is just a straight strand. Okay, it won't be in that specific double helix shape. Okay. Then describe the stage of protein synthesis which involves molecule Q. So the stage of protein synthesis involving a tRNA molecule. Anybody want to try there? So what is the process where tRNA is involved? Is it transcription or translation?
Translation, ma'am.
Correct. Okay, so now it's asking you to describe the process of translation. Okay, let's describe that. Give me a summary, or a quick summary of translation.
tRNA locates a specific amino acid in the cytoplasm using anticodons, then transfers those amino acids to the mRNA, and then they attach to the complementary nitrogenous bases.
Yes, very good. What about the proteins that it's carrying? What about the amino acids that it's carrying?
They line up in the ribosome and then protein synthesis occurs.
You're missing out one step. You're almost there. So when those tRNA molecules bring the amino acids, they drop off the amino acids, and how does it become a protein?
The amino acids are joined by polypeptide bonds, and then a polypeptide bonds, and a polypeptide chain is formed.
Okay, so as I said earlier, you need to say it's joined by peptide bonds that forms a polypeptide chain. So polypeptide means more than one peptide bond. Okay, so the bond is a peptide bond and it's forming a polypeptide chain. Okay, just correct that. Are you with me?
Yes, ma'am.
All right. Okay, so that was correct. Okay, so translation is a process whereby different amino acids are assembled to form a specific protein. Okay, so you need to define translation first. Remember, it's for 6 marks. Okay. Each tRNA in the cytoplasm carries a specific amino acid. When the anticodon on the tRNA matches the codon on the mRNA, the tRNA brings along its specific amino acid. Amino acids then become attached together by peptide bonds to form a particular polypeptide chain or protein molecule. Okay. Everybody got that? Okay, next. Now we're going on to meiosis. Study the diagram of a phase during meiosis below. Okay, identify part A.
Centriole.
Correct. Identify the phase represented in the diagram.
In the phase 1.
Yes. Actually, no. It's anaphase. It's sorry, that's not anaphase.
Metaphase, sorry, ma'am.
Yes. 1 or 2.
1.
Why would you say 1?
Sima says 2.
It's metaphase 2 because now chromatids are going to be pulled to the poles. Okay. So
Right,
that's
you're not-
metaphase 2.
Yeah, you're not seeing pairs of chromosomes at the equator. The cell equator.
Thank you.
Okay, so explain your answer. There's only it's not homologous chromosomes that are on the equator, or you can say you can just say chromosomes are lined up on the equator. Okay, explain how the phase identified in 2.5.2 contributes to variation in the gametes. Okay, and this is also a law. And now you need to explain how this contributes to variation. Any takes?
No.
Okay, so chromosomes are lined up on the equator, okay? And they are lined up randomly. Okay, so this is the law of random assortment, okay? Where it randomly aligns on the equator, which means that any chromosome could go into each of the cells. Okay, so that is how variation is produced through metaphase. Okay, the random assortment of chromosomes on the equator. Okay, how many chromosomes were present in the original cell at interphase?
Sima says 3.
3 cells. Sorry. How many chromosomes were present in the original cell at interphase?
Okay, so remember what the function of, or what happens in interphase.
Okay, any other answers? Okay, so what happens in interphase?
The cells mature.
What does that mean?
DNA replication takes place.
Correct. Thank you. Okay, so if DNA is replicating itself, that means your chromosomes are going to be double. So DNA replication doubles the genetic material so that it can be shared into the shared by the new cells, the 4 cells that's coming out of meiosis.
So, ma'am, will the answer be 92?
There's only 3 chromosomes there. I'm not sure how you're getting 92.
So
Oh,
be
my
careful
bad, my bad.
with questions like this. Unless they say in an animal cell, or if they say in the diagram given. So if they say in the diagram given, count the chromosomes there. If they say animal, then
My
you
bad.
go to the 92.
Okay, so I assume that it's in the diagram because it says study the diagram. Okay, so how many chromosomes now?
2.
So we have 3 here.
There's 3 chromosomes there.
Very
So
good. And the
remember, I just said that interphase is where the DNA replicates and doubles the genetic
So
material.
Q, you said the answer.
Okay, what was Q's answer?
After 92, he corrected it to 6.
Oh, okay, yes, that is correct. Okay, so we got 6 chromosomes at interphase. Okay, remember this is showing you meiosis. This is showing you metaphase 2. Okay, after 1 round of division. Okay, so how many chromosomes will be present in each gamete that forms at the end of meiosis? Okay, so those chromatids are now going to separate and go to the poles of that cell, and you're going to have 2 cells being formed, 2 separate cells being formed. How many chromosomes are going to be in each of those gametes at the end of meiosis?
The number will be halved, which leads to the production of gametes.
Okay, so it was already halved from going from meiosis 2 to meiosis 1. Sorry, man, what am I saying?
Meiosis 1.
Yes, it would be going from meiosis 1 to meiosis 2. The number is halved. Okay, so now we are already on meiosis 2. Okay, so now your chromosomes are going to separate and go to the poles. You're going to have your chromatids at each pole, and you're going to have cytokinesis occurring. So how many chromosomes do you end up with in each of those cells? In each gamete?
So when the chromatids separate from this metaphase, how many are going on each end during anaphase?
3.
Yes.
Correct. Okay, there's going to be 3 chromatids. Well, we'll still call them chromosomes, okay, because it's half the amount, okay, of chromosome, half the amount of DNA that goes into each gamete, okay, or 1 copy of that chromosome going into each gamete. Okay, so which process resulted in the 2 chromatids in each chromosome being different from each other? Okay, so you can see that each of these, you can see that first chromosome has a small white segment and the second chromosome has a small black segment. Okay. In fact, if you just look at the first chromosome, because it says the 2 chromatids being different from each other, okay, what process caused that? Why is there a white segment there in that 1 chromatid? What process occurred there?
Crossing over.
Yes, correct. Okay, so crossing over happened where there was swapping of the alleles, and we ended up with different chromatids on one chromosome. Okay, correct. Okay, the diagrams below represent two phases of meiosis in an organism. Identify the phase of meiosis represented in diagram 1. What phase of meiosis is represented in diagram 1?
And phase 2.
Yes, okay, correct. Identify part A.
Centriole.
Yes. Part B.
Centromere.
Correct. Part C.
Spindle fibers.
Yes. Okay. State what happens to structure D in the next phase of meiosis. Structure D. So we're looking at those two chromatids, that one chromosome there. What happens in the next phase?
They get pulled to the equator of the cell, man.
They get pulled to the equator.
Oh, sorry, like pulled away in the phase.
They are pulled to the poles of the cell. Yes.
My
Okay.
bad.
Okay. Name the process during which genetic material was exchanged as shown in the diagrams above. So when your genetic material was swapped around. We just spoke about it before. What's the process called?
Crossing over.
Yes, very good. Okay. State the consequence if the process name that you just named does not occur. So if we didn't have crossing over, what would happen?
There'd be no genetic variation.
Yes, correct. You'd have identical daughter cells. Okay. Which means your offspring would all be identical to each other. Okay, give the number of chromosomes present in the original parent cell in this organism. Okay, we did a similar question just before this. What would be the number of chromosomes present in the original parent cell? So this is meiosis 2, okay? So in meiosis 1, that is your original parent cell number of chromosomes. So what would that be? Any ideas? There's 2 chromosomes there now, and they are unpaired. So in meiosis 1, they are paired. So what is the total number of chromosomes? Students, any ideas? Yes, correct. Okay, you'd have 4 chromosomes. Okay, the number of chromosomes present in a human cell in the same phase as that shown in diagram 2. So now it's asking you for human cell. So what is the answer to that? The number of chromosomes present in a human cell in the same phase as diagram 2. What is diagram 2 showing? It is metaphase 2. Okay, so what is the number of chromosomes in metaphase 2 in humans?
Three.
Correct. Okay. Okay. Identify the phase of meiosis in the diagram above. What is the phase that's showing there?
Look at what's happening to the cell membrane.
Telophase 1.
Correct. Okay. Draw a diagram to show only the 2 gametes formed from cell A. No labels are required. See, it'll specifically tell you now whether labels are required or not. But if it doesn't tell you, then you must always just do labels. Okay. So draw a diagram to show only the 2 gametes formed from cell A. Okay. So that now will be a biological drawing, simple, clean lines. You're just going to draw a cell, a nucleus. You're going to show how many cells are you drawing, by the way? Cell A is going to split into 2 cells, 2 daughter cells, and each is going to have 1 chromosome in each. Okay, that's what you're going to draw. And you just have to draw a nucleus around them, around each of those chromosomes, exactly like what's in the diagram, just it'll be the offspring, or it'll be the gametes of cell A only. The daughter cells of cell A. Okay. Tabulate 2 differences between prophase 1 and prophase 2. Okay, what is the difference between prophase 1 and prophase 2?
So what happens in prophase 1 and what does not happen in prophase 2?
Any answers? In prophase 1, what's the major event that occurs?
I mentioned it in the previous question.
It's very important. You need to know this process. It's super important in meiosis. It's called crossing over. You must, it's going to be an answer for something, so you must know it. Okay. Crossing over causes genetic variation in the cells, and it happens in prophase 1. Okay, it only happens in prophase 1. It does not happen in prophase 2. Okay, so that is one difference. Crossing over takes place in prophase 1 and not in prophase 2. Okay, so I've given you one answer. Now give me the next answer. What is one more difference between prophase 1 and prophase 2?
In prophase 1, there are homologous pairs, and in prophase 2, there are no homologous pairs.
Correct. Okay, you can also say the cell is diploid in prophase 1 and haploid in prophase 2. Okay. Okay. Identify part C in this diagram. What is part C?
You gave this answer before.
It's the centriole.
Centromere.
Centromere, very good. Name the phase represented in the diagram.
Metaphase 1.
Correct. B represents homologous chromosomes. What are homologous chromosomes?
Chromosomes that are similar in structure.
Okay, give me more of an answer there. Similar how?
Similar in size and shape.
Correct. What else? What about the genes? It has the same sequence of genes. Okay, so it's a pair of chromosomes with the same structure or location of the centromere or the length, okay, and the same sequence of genes, okay, and one of the chromosomes is of maternal origin and the other is of paternal origin. Okay, explain the appearance of chromosomes in the diagram. Why does it look like this? See, this question keeps coming up. Why do the chromosomes look like this? Basically, it's asking why is there black and white segments on the chromosomes? Okay, so then you're going to say what occurred, what process?
Crossing over.
Yes, crossing over occurred, which did what?
Cause genetic variation.
It caused the, you can be more specific and say the genes were swapped. Okay, you can, okay, so there's a mixture of genetic material between the non-sister chromatids. Okay, so crossing over took place during prophase 1. Okay, state the function of part A in the phase following the one represented in the diagram. So what is the next phase? The next phase of this, if this is metaphase 1, what's after metaphase 1?
Anaphase 1.
And what's the function of that?
The chromosomes are pulled to the poles of the cell.
Yes, correct. Okay. The total number of DNA is 12 arbitrary units in each daughter cell at the end of the cell division. How much DNA was in the parent cell at the beginning of cell division? Okay, so again, this question is always coming up. You need to know that if the daughter cells are haploid, what is the parent cell? The parent cell is diploid. So we're going to always double the daughter cell number, okay? So if there's 12 in each daughter cell, there's going to be how many in the parent cell?
$24.
Correct. Okay. Let's do the last one quickly. Okay, identify the phase represented in diagram 1. The chromosomes are, the homologous chromosomes are on the equator. So what is that phase? You need to always check in the diagram, is it homologous chromosomes or just normal chromosomes, unpaired chromosomes? Okay. If it's homologous chromosomes, that means it's meiosis 1. If it's unpaired chromosomes, it's meiosis 2. So this is showing you homologous chromosomes, which means what phase of meiosis 1? The chromosomes are on the equator.
In the phase 1.
Correct. Sorry, no. Try again. Look at diagram 1.
Oh, metaphase now.
Yes. Okay. Now, what's the phase of showing in diagram 3?
Telophase.
Okay, telophase and cytokinesis. Okay. Give the letter only of the part that contains DNA. Which one is showing you DNA? Which one showing you DNA quickly?
A, B, C, or D, or E?
Students, you should know this answer. It's easy. The answer is B. Okay. You're looking at the it's showing, pointing to a chromatid. That is the DNA. Okay. Which letter attaches to the centromeres of the chromosomes? What's the answer?
D.
What?
Can say that one more time for us?
It's D.
Okay, so what attaches to it? Which part attaches to the centromere?
The spindle fibers. OC.
Correct. Correct. Okay. Give the letter that forms the part that forms spindle fibers.
Then you would say, Ken, your previous answer.
D.
Yes.
D. Okay, the centriole. Okay, then name the organ in a human male where meiosis occurs.
I think it's the testes.
Correct.
Correct. Okay. Okay, so we managed to get through all our questions. Well done. Okay. Hopefully everybody is more comfortable now with meiosis and the section on DNA. Please remember to learn your DNA replication, learn transcription, translation, okay? And also make sure you know meiosis and your phases extremely well. Make sure you know what is crossing over, the purpose of crossing over, okay? You need to know the ploidy or whether it's haploid or diploid in meiosis 1, meiosis 2, okay? Those are key things you need to remember, okay? so that you make sure you get your 100% of your marks in meiosis. It's very easy. Everything's 1 mark, okay, or 2 marks. So make sure that you're going to get all the marks that you can in the meiosis section. Okay, so if there's any questions, I'll take questions now.
Ma'am, may you please elaborate on NBTs and how they work?
NBTs?
Yes, ma'am, the national benchmark test when you apply for varsity.
Yes.
Oh.
So UKZN itself does not do the NBT. They normally accept you based on your trials or your final marks. I know only sometimes medical school may have it, but that they will contact you with regards to it. It's like an aptitude test that they will ask you to do.
Okay.
Jerusha, if you can stop sharing, I can play the video for them on how to apply to UKZN.
Okay, sure.
This works. It wasn't working earlier. Okay. Just know if you can see here. Oh,
no sound.
It's not coming loud. My volume is doesn't want to increase. 1 second for me, students. Zoom really freezes YouTube for me.
The extent of one's success can be attributed to those they rub shoulders with. Therefore, you must surround yourself with like-minded people. So, how do you become part of a community that is committed to your self-development? How do you become part of UKZN? First, you have to apply. All applications for undergraduate studies at UKZN are processed.
Oh, so sorry. Please.
Or maybe you can share the link for that.
Yeah. Okay.
Yeah, if you just share the link on the chat, it might work better.
Ma'am,
yes.
When you apply for medicine, which reports are required, like grade 11?
Initial applications.
Oh, sorry.
Yes, your grade 11 and your you'll have your first term, your first and second term Matric reports, because applications open in June for medicine.
So is it the grade 11 end of year and then Matric first and second term?
Yeah.
Okay, and Matric end of year, do we use that or?
Yes, you will. They will only give you a final acceptance once they have that.
So there are 4 in total that are required.
Yes, but you don't have to send them anything after you've applied. You don't have to send them your final Matric. They get it automatically.
Okay, thank you.
Yeah. Okay, students, thank you for joining us today. And I will see you at 6 p.m. for our last session on genetics. Okay, thank you very much. Have a good Saturday afternoon, and I will see you this evening. Okay. Thank you. Bye.